Hey everyone, I'm Cory Delair, and today I'm going to be talking about epigenetic gene silencing by heterochromatin in fungi and how it primes fungal resistance to antifungals in medicine today. In this talk, I'm going to be going over some background information on fungus and how fungal resistance is becoming a major problem in modern medicine. Also, how epigenetic changes or epimutations in fungal DNA can cause changes in phenotype without actually changing the genetic code. I'll also be going over a research study done by Torres Garcia, and it was a study on epigenetic changes and how it affected fungal resistance. I'm going to go over the methods, results, and their conclusion. So, just a little intro to what fungus is as an organism. It's a eukaryotic organism, and its environmental role is as a decomposer. For the most part, it breaks down organic matter in the environment and essentially returns it as nutrients into the environment to be used by other organisms. However, while they do have a positive role in the environment, they can be extremely dangerous as invasive pathogens to humans. They are harmful in a variety of organs, such as like your eyes, your brain, your skin, internal organs, and they can potentially lead to long-term effects or death if untreated, if left untreated, or if treated improperly. One major problem causing fungus today is the Candida. It is a group of fungus that is growing increasingly resistant to what we have available for antifungals. And in one specific antifungal, flucazone, which is commonly used or was commonly used at one point, 90% of Candida strains have become resistant to it. And in medicine today, there are only three classes of antifungal drugs. So we only have like three ways to kill a fungal or prevent it from getting any worse. And the classes are first pollenes, which act on the cytoplasmic membrane in an attempt to destabilize the fungus and kill it, hopefully. This is right now probably what's being used the most in medicine. There's also triazoles, which is what that flucazole was, or fluconazole was that I mentioned earlier. And it inhibits the synthesis of enzymes that the cell used to support itself and maintain homeostasis. Essentially, it's just trying to prevent it from developing any major products that it needs to survive. The major issue with this is that triazoles are used a lot, and because of this, a lot of fungus have developed resistances to this type of antifungal. And finally, there's the echinocandins, and these directly attack the cell wall in an attempt to lyse the cell walls, killing cells, hopefully killing the fungus. And the major issue with this is that it hasn't even yet been approved for the use, like of treating small children or anyone who's not considered a healthy individual, as echinocandins could potentially be toxic to humans in high concentrations. It's still being tested. Uh, how do fungi become resistant to all this? A primary cause of it is just genetic mutations. They are known to mutate rapidly as they have short lifespans. Generations pass very quickly, and this leads to a lot of changes in genetic code. But something interesting about fungus specifically is that they have the ability to change phenotype expression without actually changing their DNA. And this is done by heterochromatin mediated silencing. So what fungi can do is they can manipulate the location of heterochromatin in their DNA, meaning they can essentially relocate what they want to be read in their DNA and what they don't want to be read. They can silence certain genes and in turn silence an expression or cause a new expression of genes. And the formation of this heterochromatin is caused by H3K9 methylation, which is a histone modification at the H3 histone done by lysine 9. And it just causes the DNA to bind tightly, forming heterochromatin. With all this prior knowledge, Garcia-Torres and the rest of her lab came up with the hypothesis that if heterochromatin can relocate in wild-type cells, then epimutations could be generated that allowed the cells to adapt to external insults. Meaning, if they are really able to at will change phenotypic expression, they could adapt to any sort of external pressure like an antifungal. And if this first hypothesis is true, they then went on to say that if this is true, that these mutations are likely to be extremely unstable and will be lost after an absence of the external insult. So if these fungus are in an environment with a antifungal and they begin to develop a resistance to it, if taken out of the environment and then placed back in it, they will most likely lose their resistance. And their approach to this was by colonizing a large amount of Schizosaccharomyces pombe, otherwise known as fission yeast. They could monitor the growth of fission yeast in a caffeine-positive environment in moderate levels of caffeine over 20 days and monitor the growth. And after 4 days, about half of these would be placed in a non-caffeinated environment if they are showing that they can grow, and later to be placed back in the caffeine-positive environment. After doing this, they did notice that there were two different types of resistances that were being developed by these pombe. There's the unstable resistance, which after 14 days out of the caffeinated environment, once being placed back in, they began to slowly die off. And you can see that in this top half of the data here. And the second mode of resistance was what they would call stable resistance, and that is after 14 days of being in a non-caffeinated environment, when placed back in, they actually managed to maintain their resistance. And what they gathered from this is most likely support that something's going on, a stable genetic change had occurred in terms of genetic mutation. Whereas with unstable resistance, it most likely supported the idea that these heterochromatin locations are not disappearing, but relocating back to an original spot once taken out of the external pressure. And to view this, they did this thing called a serial dilution. That's what you see here with the black and gray pictures. So what they did is, as they were plating these fungus, in each plate, they would place less and less going from left to right. And typically when doing a serial dilution, this could be done in a factor of 10, which is really common. So say there were like 10 to the 7th, 10 to the 6th, 10 to the 5th, 10 to the 4th. And that's why you're seeing less and less fungus in each of these plates. And that just gives you different ways to look at how the caffeine is affecting it, or just a more clear picture of what's actually going on. And with the unstable versus stable mutation, one thing that was already known to them is that the CLR4 delta gene, its primary function was the formation of heterochromatin in fungi. So they came up with the idea that if they removed this gene and heterochromatin couldn't be relocated, that they would immediately lose resistance. And that's what you see here in the UR1 or unstable resistance trial 1, that The control group, once placed in a caffeinated environment, kind of maintained resistance. Some lived, some didn't. However, once uh, the CLR4 delta was removed, you can see an immediate loss of resistance. Almost everything was beginning to die out. However, with the stable resistance, once moved from the non-caffeinated environment back into the caffeinated environment, there really wasn't any changes. It clearly shows that these fungus were able to hold their resistance even without the CLR4 gene, meaning they didn't need that ability anymore to produce heterochromatin at different locations in the chromosome. And after doing all this work, they analyzed a total of 176 different colonies. And what they found was most of the time it is extremely difficult to tell what type of resistance is being developed by a certain group of fungi. And so about 64% of it was left unclear. A lot more, and you can see in screening 1 and screening 3, they clearly had a lot of trouble determining what type of phenotype was being shown. Unclear doesn't necessarily mean resistance wasn't gained. They just couldn't figure out exactly or pinpoint exactly what type it was. But as we can see, for the most part, unstable resistance is the type of resistance that was showing through in these fungi, while at the same time a good amount of stable resistant fungi were, or a good amount of the stable resistant phenotype was showing. And finally, at the very end, what they did was for the unstable and stable mutations that they found, they did genetic analysis using a genome browser for Pombe. And what they found was 80% of unstable isolates had a new heterochromatin domain at a shared locus, meaning 80% of the newly resistant fungal strains all showed that they had newly developed heterochromatin in the exact same place, which is strong evidence that the formation of this heterochromatin is turning off some type of gene that is allowing them to become resistant to the caffeine in this situation. And they later tested it with a specific antifungal, and they were getting similar results to what they saw with caffeine. They also found that genome sequencing of the unstable isolates showed no mutations in regions relevant to caffeine and drug resistance, and that 27% of them had no detectable difference in DNA sequence from the initial wild type. So these epimutated fungus genetically were the exact same as the wild type fungus that they were looking at in 20% of the instances. However, this really supports that, again, it's this change in heterochromatin location. It's relocating into a place that can allow it to live in a unstable environment or in some type of external pressure. And with these stable isolates, they actually found that there was a genetic mutation. The genetic mutation was located on the PAP1 locus for every single one of the stable. So this is most likely what is giving the stable isolates the resistance to the caffeine. And there was no detectable heterochromatin alteration in the DNA of these fungus. And just some key points I'd like for you all to get from this presentation is that fungal infections are increasingly difficult to treat because resistance to current treatment methods is a major issue in the healthcare industry. As I said earlier, we only have 3 different classes of and antifungals right now. And as things are getting more, as different strains of fungus are getting more and more resistant, it's just going to continue leading to more and more health issues. Treatments are going to continue getting worse, so something needs to be done about this. Fungal resistance can be the result of a mutation or an epimutation, with most instances being an epigenetic change. And then finally, it is possible to pinpoint the location for the formation of heterochromatin, and could potentially be useful in the development of a more effective treatment for fungal infections. So specifically with the Torres-Garcia study, what they found is that there are almost 100% regions that we can pinpoint where there are for sure being changes to the DNA made, whether it's genetic change or just a change in the structure of the, or a change in the heterochromatin locations. But if we can pinpoint where it is, we can develop more effective antifungals that can specifically target whatever is being produced by the change, and therefore making more effective antifungals. And thank you for listening to my presentation.